Is this on? It is now. Okay. I'm going to ask everybody to find their seats so we can get started with the next presenter. Great, thank you. I'm trying to keep us all on time and all on schedule so we can A, get to lunch because, you know, I don't know about you, but I get hungry, and B, to keep everybody online on track so they can get their credits as well. So right now I'd like to welcome Dr. Paul Regier. Dr. Regier is a postdoctoral researcher at the University of Pennsylvania. He's interested in the intersection of research and health policy. He has a strong interest in understanding the neuro and environmental mechanisms that contribute to mental health and addiction, and wants to leverage this information to identify optimal treatments. Currently, he's studying the effects of prior adversity and addiction on brain and behavior in humans, but he also has a preclinical background as well as a decade of experience in technical writing, data analysis, project management, literature reviews, and presentations. So welcome, Dr. Regier. Thank you.
Thank you, Erin. Thank you very much. Thank you for the invite to come and talk today. Is this better? I also recently, so an update to that is I recently got faculty at Penn, so I'm now a research assistant professor, so I'm an assistant professor on the research track. So that's very exciting. I got a K01 to study opioid use disorder and neurocognition of opioid use disorder and treatment implications. So this title is a little bit different than what's on the program. I know it was generally kind of about the neuroscience of trauma and addiction, and so we're going for a very general type of title at the when we first set this up. But this is more like hyper-focused on the neurocorrelates of cue reactivity, specifically drug cue reactivity, and then the impact of prior adversity. So we'll start with, of course, the opioid use disorder and cue reactivity. So, you know, briefly going over the opioid crisis. As everybody is well aware, just some little tidbits about that since COVID and the increases in overdoses that have occurred because of COVID, or, you know, alongside COVID. Then I'll talk about how cues work, how drug cues work, and how they get sort of imprinted in the brain. Why they're relevant, how they're relevant, what they do to our body physiologically, biologically, and then the roles of cues in addiction. And so for the second half, then I'll talk about how prior adversity interacts with drug cue vulnerability, how there's an overrepresentation, which I won't have to get into too much, thankfully, because Denise will be very much talking about the implications of prior adversity much more eloquently than I can, since she's been working in that field for decades, and I've sort of just dabbled in the last, 5 to 7 years. But there is overrepresentation in addiction. Marty kind of touched on that briefly as well. How prior adversity affects reward modulation, and then what we kind of call this adversity-driven cue vulnerable phenotype, and how that is important for implications and how to treat addiction. I don't have a lot. I know one of the bullet points is about treatment implications, and I will talk about that, but I don't have a lot to say about it because there's a lot of research that needs to be done in order to kind of get at this cue vulnerable phenotype, especially the adverse driven cue vulnerable phenotype. Okay. When okay. Just checking. Okay, some of these slides are a little screwy, but no worries. So when we, since 2017, we kind of hit a plateau up until about the COVID pandemic. And so the first time I gave a variation of this talk was in June or July 2020, right at the beginning of the pandemic. And at that time, our overdose deaths were around 72,000. And then in about 3 months' time, we saw this spike, about 18% increase in overdoses, mostly associated with opioids. About 75% of overdose deaths are associated with opioids, as most of you are probably aware. And so at that time, if we had that same increase, the 18%, if that had stayed stable across a year, we would have ended up with around 85,000 overdose deaths. What actually happened is that we ended up having a 30% increase that first year. And then we had another total of 48%. So almost a 50% increase since COVID in overdose deaths. And apologize for the slides. They're getting, they're a little changed, morphed since they are in this version, but about a 50% increase in overdose deaths. So a clear problem. It's still clear, still a problem. And these, and it's only half the story. Because as you know, many people come in with overdoses, non-fatal overdoses, about 1 million per year. And if you have an overdose, that increases your odds of going on to have a fatal overdose. And so one of the things that we want to do is find people who are vulnerable to relapse, even after an overdose. So people, have this dire, situation where they almost die because of an overdose. They come back and they go on to continue to use overdose or use opioids or drugs or whatever the case is. And so we're really interested in that phenotype, that vulnerable phenotype, the relapse vulnerable phenotype, and what's going on in the brain. So to talk about that, one of the main pieces that's driving, drug-seeking behavior are drug-related cues. So these cues are something that cause like increased desire and craving to use drugs. They have a power motivational effect to go out and seek drugs. If anybody has been a smoker in this room, I used to be a smoker, and the sights and sounds of smoking still get to me even after all this time. And they have a very powerful pull, these cues associated with drugs that cause a person to just kind of have this increased motivation to go out and seek. So how does that work in the brain? Well, we'll start with Pavlovian conditioning. So if anybody studied psychology in this room, which I'm sure a lot of you have, you're aware of Pavlovian conditioning. Here is one of his dogs that he used to measure salival responses when dogs drool. And he was measuring saliva responses to things like food. And so what happens in a normal situation is that there's something called an unconditioned stimulus, and this can be something like food or something that causes arousal, like sexually related, or something aversive related, causes an unconditioned response internally. And that can be expressed externally as well. So that when a dog sees food and is hungry, the dog will drool. And then the other piece of this that's important is the neutral stimulus. And so a neutral stimulus doesn't necessarily signal anything at this point. And the dog just might be like, okay, that's a bell, so what? Don't care. But what happens is that a neutral stimulus paired with an unconditioned stimulus like food that causes drooling can go on to take on the properties of food. So as long as it's paired with this food or this stimulus that causes or elicits a response consistently, it will take on the properties, in a sense, of that stimulus and cause a conditioned response. So this response is now conditioned to respond to this what was once a neutral stimulus, like a tone or a bell. And that's what Pavlov used, a bell. He rang it right before he presented food, and then the dogs would drool. And after enough time, he saw that the bell, when he rang it, would elicit drooling. So how does this work in the brain? Well, there's some studies by Wolf and Schultz in the late 90s and ongoing as well. But these were the sort of seminal studies where he was looking at monkeys and he would put an electrode into their brain, into the middle brain, the ventral tegmental area, and he would record what was dopamine neurons in that region. And these are neurons that project out to several different parts of the brain, like the nucleus accumbens, which we all kind of associate with reward. It's a little more complicated than that, but it's fine to think of it as like reward motivation projects to like the amygdala, prefrontal cortex, places like that. And so this researcher, Wolfram Schultz, he would give the monkeys juice, and then he would measure dopamine response in different conditions. And so here's a schematic of the brain. So he's recording from this area here, the midbrain, which includes the ventral tegmental area. And as I said, kind of projects out to all these different regions in the brain. We're kind of focused right now on this midbrain area. And so what he, so like I said, he would give a juice reward, and he would measure the dopamine response. So this blue line is representing a tonic state of dopamine. And if you see an increase, that is just a representation of dopamine activity increasing in response to, you know, some stimulus, or in this case, reward. When you give a natural reward to an animal or a person, and it's salient or highly palatable, you'll see an increase in dopamine response to that reward. So not too surprising. But what he did is that, like Pavlov, he started to pair different stimuli with the reward. And so he would pair like a light to, he would like show a light and then give the juice, and watch what happens in the brain, watch the dopamine response in the brain. So at first, the light doesn't have any conditioned properties, so brain doesn't really care about it one way or another, it's neutral. But after a while, if you pair it consistently with a consistent amount of juice, that light cue will take on some of those properties. So it'll start to predict the reward about to come. And then after a while, you see more dopamine activity responding to the light and less to the reward. So eventually what happens is that the light cue will elicit a dopamine response, but the juice won't. So what is happening there? It seems that, I mean, the theory goes that the light cue is, like I said, kind of taking on the properties, and that dopamine response is back propagating to the light cue, so that it is predicting the reward. And so as long as this relationship stays consistent, this dopamine pattern won't change. You'll see a response to the light cue, and then you'll see this tonic response to the drink, but no increase or decrease. However, if you do change the reward, so this light has been predicting a certain amount of juice occurring, if you take away that reward, then all of a sudden dopamine says, where's my reward? This used to be where the reward was happening, but it's no longer there. So you see a decrease, a dampening in response. And sometimes a decrease in reward can also lead to feelings of like dysphoria or negative type of affect. So this is sort of like, we'll get into it, but if you see a cue, like if you were a smoker and you saw someone smoking, but you didn't smoke, you might have this craving, or you might have this sort of dysphoric feeling associated with that cue because you didn't get the reward. You're hoping to get that reward. Your brain is predicting that you'll get that reward, but you don't get it. Similarly, if you increase the magnitude of reward, you'll start to see a dopamine response to reward again. And as long as this pairing stays consistent, that again will sort of backpropagate to that stimulus, and it will just take on that property. It will predict 2 glasses of juice rather than 1. And to kind of make this more relevant to how humans use drugs and take rewards is that there can be multiple stimuli that can predict reward. So in the first example I was showing is that you have a light cue that was predicting reward, and that would take on those properties and it would elicit dopamine, but the reward would not. You can also pair a cue with another cue so that that first cue predicts the next cue, which then predicts the reward. And as long as those all stay consistent, this is a relationship that you see in the brain. And so this is basically laying out a path. This is an evolutionary type of neurological activity that allows us to survive and find food in the environment, for instance. So it's this natural pathway, a learning pathway that shows us there's food over here by that tree, and then you start to associate the tree with reward, and then you start to associate the surrounding meadow or whatever the case may be, maybe certain flowers, whatever it is, with that reward. So as long as you see the flowers, you might see the tree, then you see the food. It's just an evolutionary type of response that allows us to survive. And so with our schematic, it might look something like this. You predict a tone, use a tone cue to predict a light cue to predict a drink. So in a human, that might look something like this, where you have the drug taking behavior. Initially, this is something that causes the dopamine response to increase. The preparation or the sights and smells of the drug might then take on those properties of dopamine and predict the coming reward. And then even the context itself can predict reward as well. So the room where you typically use drugs will elicit dopamine response, which is predicting you preparing the drug, which then goes on to, you go on to use the drug. So why is this all relevant? As I mentioned, they show how drugs might lay out a path, and that context matters, and that paraphernalia and other drug-associated items matter as well. And it also shows us why things like drugs and drug-related stimuli can be so powerful for a person who uses drugs or who has used drugs and is still having problems with relapse, that just the environment around them where they used to use drugs or associations with drugs can cause this internal motivation to go out and seek them themselves. So why are they relevant? In animal studies, we use animal models of relapse called self-administration studies. In these studies, investigators will put a rat into a chamber that has a lever and light cues and things like that, and implant a catheter directly into their jugular vein so that when they press a lever for cocaine or opioids, they'll press a lever, they'll receive the drug, receive cocaine or opioids. And rats will readily learn to self-administer drugs in this way. You have to train them a little bit, put a little bit of peanut butter on the lever, get them interested in the lever, but eventually they learn that they can press that lever and get drugs. And so rats will learn to administer heroin, they'll learn to self-administer cocaine and nicotine. They'll learn to essentially self-administer lots of different drugs of quote unquote abuse that we study and that we see people have problems with. And then after they learn to take the drug in this manner, then you take away those contextual cues and you turn off the pump so they can't get drugs anymore via the lever. And they learn then, this is called extinction, that the lever doesn't have any drug related properties anymore. So after they have relearned that the lever doesn't mean anything anymore, you can then use different sort of stimuli or drugs or what have you to reinstate that lever seeking behavior. And so this is shown with, you can give cocaine, amphetamines, heroin, and even cues or context to reinstate that drug-seeking behavior. And so this study shows nicely that context has a big part to play. And I think this is really important for human drug taking, is the context in which we find ourselves in when we're using drugs can elicit that sort of like motivation, that desire to go out and use. So what they did is that they trained rats to self-administer in one context or another. And so each context had different types of stimuli. In context A, for example, they had different sizes of box, they had different stimuli on the wall, like circles on the wall or squares on the wall or stripes. They had different color house lights, even different filling floors, and even olfactory smells in the chamber. So they trained them in one chamber, and then they extinguished them in the other chamber. So they took them out of the first chamber and they put them into the second chamber. And they quickly relearned that the lever doesn't really have any drug properties anymore. And what they could do then is that they could either turn back on the cues that were associated with self-administration in the first place, and that would reinstate behavior, or they could just put them back into the original context in which they learned to take drugs, and that would reinstate behavior. And so you can see here that they learned to self-administer drugs. This is lever presses on the y-axis, and this is just number of days. So they would sort of maintain a certain level of responding for a certain number of days, and then they would take them out of the chamber in this portion here, and put them in the other chamber, turn off the lights and the cues, and you can see their lever responding going down. And then when they put them back in the other context, or they turn back on the lights, you can see the increase in drug responding. And so why, besides the sort of drug seeking or motivation that occurs in response to these drug cues, why else might this be important? There's literature out there stemming even way back into the 1990s with Chuck O'Brien, who found that these cues and environmental stimuli had drug opposite effects. So you can present someone with this apparatus that was essentially like you're preparing the drug. You're not actually taking the drug, but you're preparing the drug. You sort of fake inject yourself. And they would see this physiological response that was mimicking, was opposite to what the drug would do. And so they found that essentially these cues would then prep the body for the drug, and then it would have the opposite effects of taking the drug, so that the drugs wouldn't have as much of an effect in their body physiologically. And so this is, part of tolerance. But tolerance seems to be somewhat based off of the context in which you are in, which is why some studies have found that even though people are taking drugs in a certain amount that they might normally take, but they're in a different context, they can overdose. So taking the same amount of drug in a different context, they overdose. And the only reason that would be is that you have, one of the reasons that would be is that you have these drug opposite effects that are occurring and causing tolerance, or acting against the actual drug reward that you're receiving. Thank you. Okay. So cues are also, as I kind of mentioned, sort of been saying that these cues will cause drug-seeking behavior, but that's just me saying it. So there's literature out there showing that drug cues will elicit craving. So these studies are showing craving here, and as you show cocaine cues over about 30 minutes, they saw a positive correlation of watching the cocaine cues with increased craving. And they didn't see any increase in craving associated with neutral cues. So it wasn't just that they were spent enough time away from the drug. It was the actual viewing of the drug cues that caused the craving. And studies have looked at in the brain where this is occurring, so drug elicited neural responses in human brains using fMRI. And so these were drug cues, and I think mostly cocaine using cues, since this was the late 90s, and most of the research that we were doing at that time was in cocaine use disorder. And so they would show drug cues, and they saw a heightened response in the striatal regions. And the striatum is a place which receives a lot of dopamine activity. And so not too surprising that they would see an increase in a response there, reactivity there in those regions. And importantly, they found that this increased response in the brain was associated with increased craving as well. So if they had more craving after the session, they also had more, that was associated with more reactivity in striatal brain regions. And a recent review, it's been somewhat controversial whether drug cues and craving are actually good predictors of future drug use or relapse. But a recent meta-analysis came out looking at about 18,000 records, 51,000 participants, and saw that indeed increases in cue and craving were associated with more than double the odds of future drug use or relapse. So in fact, with this meta-analysis, it's just one meta-analysis, but with a lot of different records showing that craving is associated with future drug use. And this relationship of increased craving and future drug use was mediated by cues. So cues had a large part to play in that association. And this is just also showing more regions that are associated with cue-induced cocaine craving. So the amygdala is one of those areas that receives a lot of dopaminergic projections. And they saw increases in the amygdala to drug cues, and that was also associated with increased craving. And so other regions too, that we don't necessarily have to get into, but like the amygdala, temporal regions, orbital frontal cortex, all are important regions, it seems, for cue-induced drug craving. And so we know that the craving, or at least we have evidence that the craving itself can be associated with future drug use. But what about the brain response? We know that it's associated with craving, and we know craving is associated with future outcomes, but the relationship of brain reactivity to drug cues and future outcomes is much more mixed. So it's not clear, and we probably just don't have enough studies, enough people, but the brain's response, as we've been studying it, has not been that predictive of future outcomes. There's some studies that show that it is, there's some studies that show that it isn't. So it's a mixed bag. But a study that we did somewhat recently, we were looking at individuals with cocaine use disorder, and instead of looking at just the overall brain response to drug cues, we were looking at the temporal dynamics of the brain's response to drug cues. So what does that mean? I'll get into it. But we were looking at cocaine use disorder, and we were looking at 73 individuals. Most of them were males who tend to be the primary population of individuals who smoke cocaine in Philadelphia. So we tend to get mostly males. And we looked at 3 different clinical outcome groups. And these may not seem like great outcome groups, but this is the best that we have with this population. So we tend to get a pretty severe population that comes in for these studies. But we had our best group, our good outcome group, were individuals who had less than 40% cocaine positive urines over a course of about 8 weeks. So less than 40%, not great, but that's the best that we could get. And then we had a lot of individuals who had greater than 85% cocaine positive urines over a period of 8 weeks, and then everybody in between. So the less than 40 group was 15, the greater than 85 was 34, and in between was 24. So there's some individuals that are doing okay after they get, start in the study, but most of them are not doing as well. This is just to show that we didn't see any major significant differences between the groups, so no confounds to worry about. The only thing that we did see was that cannabis use was higher there was a significant, I should say, there's a significant difference of a one-way ANOVA between these three groups, and the in-between group was showing the least amount of cannabis use. I'm sorry, had that wrong. So the greater than 85 group was showing the most amount of cannabis use, but we controlled for that in the analysis and it didn't change our results. So our study was, this is our study design. We were showing novel evocative cues in the first half of the task, and then we would repeat those cues in the second half of the task in pseudo-random order. So we could look at how the brain was responding initially to novel cues, at least novel to them. Some of them are drug-specific cues, and some were sexual cues, some were aversive cues, and some were neutral cues. And so this is just showing some of the order, not super important, but essentially showing that, here's one order of how we're showing the cues, and then the second half, they're not seeing them in the exact same order. It's pseudo-random. We just don't want like more than 3 cues shown at the same time. And they were shown at 500 milliseconds per cue with 1500 millisecond interstimulus intervals. And so they would see 24 of each of the different cues in the first half, and then the same 24 of each in the second half. And so what we found was that we found an interaction of group by half. In this case, half means novel versus repeated cues. And we saw that in several different areas, including the amygdala, hippocampus, midbrain, parahippocampus, and some other regions not necessarily associated with mesolimbic regions like the fusiform, which responds to like facial stimuli and other emotional type of stimuli, and the posterior cingulate cortex. So what we found is that those with the greater than 85% groups, so we'll call them the worst outcome group. The worst outcome group is shown in red, and their response to cues initially did not differ from those who had the better outcome group. But they tended to sustain their response to repeated cues in the second half. So they had a harder time sort of extinguishing the brain response to these cues in the second half. Even though they saw them once and they didn't predict any sort of reward, in the second half, they were still responding to them significantly more than those who had better outcomes, who actually reduced their response to repeated cues. So this is a fairly well-known phenomena in the amygdala, which is habituation of the amygdala response. And those who tend to have a non-habituating amygdala response have more pathology, at least some types of pathology, such as anxiety, tends to coincide with things like prior adversity, PTSD. And so we're finding that this might also be the case in people with substance use disorders. And it's not too surprising, given that the cues, drug cues, are such an important part of a person, someone who has substance use disorder, and their inability to inhibit their either behavioral or neural response to these cues. And so those, we then sort of expect then that those who, and this of course is a small sample size, it's only 14 people, but those who can actually inhibit the response to repeated cues might go on to have better drug use outcomes. So this could be a treatment target, one where we're either training people to look at cues and have maybe neurofeedback or something like that, to then reduce their emotional and brain response to these type of cues. And it's something that It sounds like you're getting TMS. I talked to Adam briefly before this talk, and getting TMS in here and looking at those cues and also maybe getting feedback on what your brain is doing might be really valuable in showing that you can actually have control over your brain's response to cues. It just might take some practice and some time for certain people. But if you can do that, if you can habituate your brain to repeated drug cues, that might be beneficial. Okay. All right, so how does prior adversity factor into this? Well, there's an overrepresentation of prior adversity in addiction. Like I said, you'll get more of this in the coming talks. So I'll just briefly mention that those who have substance use disorders, and you've probably all seen this in your work here, tend to have more adverse childhood experiences, for example, like emotional, physical, sexual abuse or neglect, different types of like familial conflicts. And so these are just a few sort of like large sample studies showing the prevalence of abuse in the substance use disorder population. It can vary greatly from anywhere from 30%, depending on the population, all the way up to 70%, especially when you're looking at sexual abuse, which tends to be higher in those who are women, and physical abuse tends to be higher in those who are men. And then having adverse childhood experiences increases your risk, as Marty alluded to in his talk, increases your risk of developing different types of disorders, increase your risk of even trying drugs, increase your risk of injecting drugs greatly, almost 7 times as much, and increase your risk of having alcohol use disorder. So one of the reasons this might be is that prior adversity tends to interact with the dopamine system, or our motivational reward system. So one of the studies that showed this was a preclinical study in rats. And they used social isolation as an animal model of prior adversity. So there's not a lot of things that you can do to rats in order to model different types of prior adversity. But one of them is to isolate them basically at birth, which is highly stressful. And it increases the activity in their stress systems way more than those who are group housed. So there are 2 different models here, are those who are socially isolated and those who are group housed. And what they saw is that those who were socially isolated, when you evoke dopamine response via electrical stimulation in the midbrain, you end up getting this heightened response. You can kind of see the peak here is greater, the black bar, or the black line, I'm sorry, is greater than the gray line. And that's in those who are socially isolated. So it seems like the dopamine system might be primed in a way to over-respond to certain stimuli. And this is probably more relevant for things like cocaine use disorder or other stimulants, since those act directly on striatal, like dopaminergic striatal neurons. They're increasing dopamine right at the site. Whereas other drugs like opioids and alcohol, they have more indirect effects of increasing dopamine. So some of this, just grain of salt, quick sidebar, might be more important for stimulants. But they saw this both in the nucleus accumbens shell and in the nucleus accumbens core. You can see that dopamine release is higher in those who are socially isolated, and that uptake was also higher. Thank you. And that also relates or translates over to behavioral outcomes as well. So those who had were animal models of prior adversity, like they were socially isolated, tended to acquire drugs seeking behavior or drug self-administration behavior more so than those who were group housed. And so you can see that in both these examples. And then in humans, using the childhood trauma questionnaire, which is a total score of those of 5 different types of trauma, including emotional, physical, sexual abuse, and emotional and physical neglect. So they take the total score and they looked at the amygdala response to emotional stimuli and saw that those with higher childhood trauma questionnaire scores had a higher response in this amygdala response, so an over response to these cues. And these were in non-drug using populations. So just in the healthy population, their reward motivation system might just be kind of overactive, ready to respond much more to emotional stimuli. And for the sake of time and complexity, I'll just move on to our study that measured drug cue response in, again, individuals with cocaine use disorder. And we asked whether those who had some sort of prior adversity differed in their brain response to different types of emotional cues compared to those without prior adversity. And we're using, this is data that was already collected when I got into the lab. So it was somewhat of a blunt instrument. We use the addiction severity index, and they have a question in there. They have 3 questions in there about emotional, physical, and sexual abuse. So just kind of straight out ask them, like, have you ever been abused emotionally? Then it gives an example. Have you ever been abused sexually? And it gives an example. So not the best measure of prior adversity, but it was the best that we had. And it still revealed different brain signatures. And again, here's our table showing that there was no differences in key demographic areas or drug use between those without abuse and those with abuse. Do I have the numbers there? So those with abuse, and this again shows the overrepresentation of prior adversity in these samples. Those with prior adversity was 40 versus those without were 28. So it was about 60% of the population having some form of prior adversity. And here you can see the breakdown of any type of abuse, emotional, physical, sexual, or 2 or more. So same paradigm, just showing as a reminder, we were showing novel cues in the first half, repeated cues in the second half, and here's some example ones. Another quick sidebar. These are much more explicit that I'm showing. Didn't think it was appropriate, but highly explicit sexual pictures and highly explicit aversive pictures. And aversive will come into play a little bit in the next few slides. So that's why I mentioned that. But the aversive pictures are, they're like wartime photos and really kind of just really nasty stuff, not pleasant to look at all. But we use them as a way to say that these should elicit some sort of emotional response internally in a person. And so when we looked at the brain, what we saw, and these are drug cue responses in the brain. So when we looked at everybody, the entirety n equals 68, we saw increases of brain response in subcortical regions in the posterior cingulate cortex. But when we looked at the abuse population, or the ones with prior adversity, they were mostly making most of the reactivity. So their response was much higher than those who didn't have any sort of prior adversity. So pretty striking difference between the two. And then, of course, there was, when we compared the two, there was a greater response in those with prior adversity compared to those without prior adversity in regions like the thalamus and the caudate. So that was a pretty striking finding, is that it seemed like there was maybe this, like I kind of alluded to at the beginning, is that there might be an adverse driven cue vulnerable phenotype that might benefit more from certain types of treatment that are targeting this cue response. Okay. And this is just showing some of the temporal dynamics associated with our study, just to follow on what we had talked about. I won't go into too much detail, but essentially there seems to be two different types of temporal dynamics occurring. One is a sustained response to repeated cues. What we're showing here is the abuse population has, they don't differ in the first half, but then they differ in the second half because they're sustaining the response to cues, and the other population is decreasing their response to cues. Whereas you might have this initial response that's much higher in one population, let's say the cue vulnerable population, that decreases rapidly. So it's not as clear cut as just saying that if a person has a sustained response or a decreasing response that you can just target that. It might be specific to like having specific areas that you have to target, or targeting like prefrontal areas to be regulators of this response. And then finally, to combine the two, we also did a study recently in those with cannabis use disorder. And we looked at those with prior trauma and those without prior trauma. And we looked at the temporal dynamics of cue response in those individuals. Again, this is just to show that there weren't any significant differences between the two. We had 18 who had prior trauma, and we had 15 without prior trauma. So again, an overrepresentation, more than 50% having some form of trauma. And we saw a similar response to what we've seen before, is that those with trauma seem to be sustaining the response or even increasing the response to repeated cues. And this is all aversive cues this time, while the ones without prior trauma are decreasing in the response. And we saw a significant correlation with craving in this population, in that those with prior trauma who had a greater response in the second half also had greater craving associated with that increased response, whereas those without trauma didn't have that relationship. And this is just some pilot data showing that the takeaway from this particular slide is that when we look across drug-using populations, drug cues might not be the best indicator of our cue vulnerable phenotype, but aversive cues might. So all of these individuals with prior adversity, whether they had cocaine use disorder, cannabis use disorder, or opioid use disorder, they all had an increased reactivity. Increased reactivity to aversive cues in relevant brain regions like the midbrain, amygdala, striatum, and insula. But for drug cues, which I'm not showing here, but it was more consistent in those with cocaine use disorder, but you didn't see that in the other ones. So aversive cues might even be a better probe for what's happening emotionally in a person when they see these evocative cues. That's the main takeaway for that. So I've talked a lot about a lot of different things. We talked about the opioid crisis, how it's been going since COVID, how cues work, how they get embedded in the brain, the role of cues in addiction. We talked about the overrepresentation in addiction and reward modulation, and the adverse cue vulnerable phenotype. And I did promise some implications for treatment, but I can leave those for potential questions if you have them. The unfortunate thing is that it's sort of new to be looking at the brain in relation to prior adversity and its reactivity to different cues. So we don't have a lot of good data. We have some pilot data showing that maybe some of these different medications might be useful for different populations and reducing the brain response, but there's still sort of like, this is needed future work to be done, is to find potential probes or potential medication candidates to decrease this cue vulnerable phenotype. So, okay, and that is all I have. Thank you.
Thank you so much. That was Actually, really fascinating. I have tons of questions, but I'm going to leave it for, I'll start with the one online there, I guess.
That's more of a comment.
That's more of a comment. Okay. All right. Any questions? Yes. Whoops. The microphone is coming.
Thank you. Has anybody looked at drug dreams, whether they're cues or averse cues? Patients will wake up and they'll be, when I talk to them about their drug dream, that they'll wake up in a panic or in a fear, they'll be in a cold sweat. And yet you would think, when I talk to them, what was the dream? I was dreaming that I was back in where I used to use, which would be normally a regular cue. So has there ever been any thought? Kind of difficult to study because you don't know when they're going to have the dream, but just wondering.
Really interesting. I hadn't even thought about like drug using dreams, but it's pretty common then for people to have drug using dreams. Yeah, I hadn't thought of it, but it would be really interesting to have a sleep study involving substance use disorder participants. And as they wake up, you could do something, but yeah, that sounds like a tricky thing to take care of. So I'm not sure. I'd have to look that up. Thanks.
I've seen different statistics on this, and I just wanted to know if you have any clarification. When a brain is tested through usually an MRI, showing it lighting up for a craving when they're shown either drugs, paraphernalia, whatever, that there seems to be a timeline where when they get back in the machine after so many, what I've read is 12 to 18 months, where you will then retest them, show the paraphernalia or whatever, and the brain does not light up so much in those areas. Do you find that, do you have any evidence of that, or have you seen that? Is that true? Because I think that is, it's important to know, like, how triggered someone is actually getting in their brain when they see, something that could cause them a problem.
So these would be people who are abstinent for about 12 months? Yeah. I would imagine that it would happen a lot sooner than that. Going off of, different like rodent studies or preclinical studies, this happens in a matter of days, where you can learn that a certain cue isn't associated with taking the drug. So it should be decreased after, weeks to months, essentially, rather than, I would say, a full year. I don't have the exact data on that, but there is, like Rita Goldstein does work with incubation of craving. So she is very much interested in whether after a certain amount of abstinence, the craving actually gets stronger. And so there's some like controversy there, whether that would actually be worse, 6 months out versus completely gone. And I think that would have to do with like your mindset of like, you see these cues out in the environment, how do you react to them? I would imagine that if you see these cues in the environment and then you don't go use drugs after, weeks to months, the sort of internal motivational drive would be much less. However, what if you're completely avoiding all of those things? If you're completely avoiding those things, you might then one day run into that cue, and that would probably still trigger a response, even though you haven't been using for a certain number of months. So there is some need to sort of like test this out in the real world. This is all like lab-based things, so we don't have the exact answers, and there's some theories. So I think that's about the best answer I can give you. Okay.
So I guess my question relates to the adversity, which was really fascinating. So with the people with adversity in their history, they did not show the responses to the drug-related cues that you would expect. They saw it more to the aversive Is that, and the ones versus the ones who had no history? Did the ones who had a history of, no history of adversity react to the drug cues then?
They didn't really, I would have to go back, but the ones who had no adversity, they had some response. I was showing that a somewhat higher threshold just because the response in those with adversity was so high. But they were showing some response, some increase to drug cues, just not as great as those who had prior adversity. But that wasn't consistent across drug use types. So those with cannabis use disorder didn't really have a heightened response. Those with cannabis use disorder and prior adversity didn't have a heightened response to drug cues, but they did to aversive cues. And that was the same for opioid cues as well. And it might just be because the drugs like cocaine and other stimulants, they directly increase dopamine at those striatal sites or those mesolimbic sites, whereas like opioids and cannabis and alcohol kind of have indirect effect of increasing dopamine. So there might be something about that, but that's still kind of unclear to me.
Yes.
Question on the impact of anti-craving meds. Were there any folks who were on naltrexone, Suboxone in the study, or what would be your theory if folks were?
Yeah. Yeah, so he was saying, would you encourage someone to go on MAT to decrease some of those risk factors? And in response, like, were people on naltrexone or Suboxone? So for the cocaine use studies, nobody was on any medication at all. For the opioid studies, they got that scan prior to going on some form of MAT. So nobody at the time of scan was on anything. But to that point, so implications for treatment, I think, and some of the theories we have around this adverse driven Q vulnerable phenotype is that because of the interaction with the stress system and the dopamine system, that something like buprenorphine, especially for opioid use patients, might be really useful because it has that kappa antagonist properties. And kappa is so strongly associated with like negative affect and dysphoria in your system that if you're decreasing that, that might actually help decrease cravings in these particular individuals. And there's some indications that maybe a GABA B type of agonist might work too. And that's more working like top down. So you have increased regulatory responses that are dampening the response to specific drugs. So that might be helpful for like anti-relapse. And then the final one was that, as was mentioned before, like TMS, bolstering that top-down regulatory system while showing cues might be the best option, because then you're sort of reevaluating or reassessing what those cues mean to you, and you can show that you have cortical control over there. Essentially like training your brain that these cues don't mean anything anymore. I mean, that's hard to do, but the brain is over responding to them because they've had such importance in that person's life. And then you have to retrain the brain that they don't mean anything anymore. And that's a hard thing to do. So like something like TMS and neurofeedback might be a good way to do that. Or FNIRS.
Or FNIRS.
Yeah.
Okay. I want to thank you so much. I really appreciate you coming out. One more round of applause. I'd like to thank all of our speakers this morning for coming and giving such really interesting presentations. And Paul, I wish I had time to pick your brain even more. So I'll have to do it later. So before we break for lunch, if you would like a brief driving tour of our beautiful campus, please find Adam Lush in the lobby, who will take you to the shuttle and be your tour guide. And he will bring you to lunch after that. Lunch will be held across the street in the NEAG Medical Center on the bottom floor. Jess, Maggie, and Deb will walk you over, and they're all out in the hallway. I'll also be walking over, so you're welcome to follow me. And please be back here by 1:20 p.m. when we will start our next presentation. Thanks so much. Enjoy lunch. And don't get that lunch coma, because we have some good speakers coming up this afternoon. I mean,